This is the second lecture video for protein synthesis. So as a quick review, we went over transcription in the first video. So for transcription, it occurs in the nucleus of the cell, and the process is creating a strand of RNA from a DNA template. Now, the RNA that is produced from the DNA has to go through RNA processing in order to produce messenger RNA. Now, translation begins after the messenger RNA leaves the nucleus and enters the cytoplasm. So just a few other notes about this entire process. Remember that it goes DNA to RNA to protein. So your genes in your DNA code for proteins. Now, transcription is producing an RNA for a gene, not the entire strand of DNA. Now, translation, you will need to be able to identify where in the cell it occurs, describe the function of translation. You need to be able to identify the different types of RNA that are used during the process. You will need to explain what a codon is, an anticodon, and which RNA you can find an anticodon. You will also need to be able to explain the three steps in translation and how we use the genetic code table, and really what is the genetic code. So let's get started. So this is the image that I showed you in the first lecture video, what we call the central dogma of molecular biology, DNA to RNA to protein. DNA to RNA is transcription, RNA to protein is translation. So where does translation occur in the cell? Well, it occurs in the cytoplasm. Transcription is in the nucleus, translation is in the cytoplasm. So what is the function of translation? Try to move this bar here. So the function of translation is to use the information in RNA to produce a polypeptide, which is a sequence of amino acids that form a protein. I'm going to write that down for you. So the function of translation, it is use of RNA, and it's actually more than one type of RNA, and we'll get into that in a minute. So the use of RNA to produce a protein. That is like a very simplified definition of translation. That's essentially what it is, right? Transcription, you are taking DNA to make RNA. Translation, you are using that RNA to make a protein. Now, first, before we get into the steps of translation, there are a few things that you need to understand about messenger RNA. So messenger RNA is read in triplets that we call codons. And what I mean here is that the messenger RNA is read 3 nucleotides at a time, and it is these sets of 3 nucleotides that code for a specific amino acid in the polypeptide chain. I know that seems a little confusing, but hopefully as we go through these slides, it will make a little bit more sense. So let me write that down for you here. So I'm going to write a sequence. A sequence of 3 nucleotides, and this is what we call a codon. And messenger RNA is used to code for a specific amino acid in the polypeptide. Okay, so if you look at the picture here, the red line is drawn between, so you have AUG, then there's like a divider, ACG, a divider, GAG. So it is the codon, so AUG is a codon, ACG is a codon, GAG is a codon, and these codons specify the amino acids that will come along in the chain, the polypeptide chain for the protein. So this is what we call the genetic code. Now, basically, the genetic code gives you all the different codons and which amino acid they code for or specify. So there are 64 different codons in messenger RNA. Now, 61 of those codons code for specific amino acids. 3 of those codons are what we call stop codons. We will get into what a stop codon is in a little while, but here you can see it says stop, stop, stop. Those are your 3 stop codons. You do not need to know UAA is a stop codon, UAG is a stop codon. Just know that there are 3 different stop codons. And basically what those do is they show the like ribosomal RNA, they basically indicate when the protein is done, right? The end of the polypeptide chain. Now, the genetic code table, or the genetic code, is the same for all living organisms. And I want you to take a second and think about why that would be true. So it doesn't matter if you are a plant or a human or, I don't know, a pig, right? The codons in the messenger RNA code for the exact same amino acids in all these different organisms. So if a human has a codon that is UUU, it codes for the same amino acid phenylalanine as UUU codes for in a plant or a pig or anything else. Why do you think that the genetic code is the same in all living organisms? So this actually goes back to evolution. We all evolved from that first cell, right? That first cell that originated on Earth. All living organisms came from that first cell. Now, the genetic code and DNA, that has remained unchanged throughout evolutionary history. And so this is one huge piece of evidence that we all evolved from that first living cell. Okay, so what I want to do here is I am going to give you a strand of messenger RNA, and we will look at how that specifies the amino acid sequence. So let's see, I don't have a ton of room, but let's do this. So this is going to be the strand of DNA that I just drew here. I'm going to write DNA right here. Okay. Now I am going to write down a nucleotide sequence on this DNA. Now I have to keep doing a different text box for each base because otherwise it'll squish them all together when I go to click off of it, I learned. So we have A, T, C. Yeah, let's do a C here. I'm just kind of making this up as I go. I don't really know ahead of time what amino acid it will code for. And how about another A? Okay. Oops, I need one more base in there. Let's see. I think I will put an A. All right, so take a second here, look at this sequence, the nucleotide sequence here I gave you for DNA. What I would like you to do is transcribe that into messenger RNA. So ideally, we'll pause my video and do a little transcription there. Okay, so this white line is going to be my messenger RNA. Okay. So after transcription, we will have, I'm going to do this in white. So adenine in DNA pairs with uracil in RNA. Remember, there is no thymine in RNA. Thymine pairs with adenine. Cytosine with guanine. Adenine with uracil. So you should be getting the hang of this by now. Guanine with cytosine. Guanine with cytosine, and adenine with uracil. We'll move that uracil over just a little. There we go. Okay. Now remember that the whole point of what I'm showing you here is to determine the amino acid sequence, right? So DNA to messenger RNA is transcription. And I did the transcription following complementary base pairing rules. So now we are going to determine which amino acids the messenger RNA codes for. So remember that we are looking at codons, sequences of 3 nucleotides. So we have UAG, and then I'm going to draw my line because that is the first codon. GUA, second codon. CCU, third codon. Now let's look at the genetic code table on the right. You can see here, I need to do that in black. It says first letter. On the top, it says second letter. And on the side on the right hand here, it says third letter. Those are referring to your codon. First letter in your codon, second letter in your codon, third letter in the codon. I'm going to erase those arrows there. So let's look at my first codon. It is UAG. So what you want to do is go to your genetic code table where it says first letter. Our first letter is U. So right here. Whoop. Hold on one second. That did not do what I wanted it to. Okay. U. That is our first letter. So you go along that left side, you find U. Now, that means that our amino acid is going to be in this column right here. I meant row, sorry. Rows go horizontal, columns are vertical. So U is our first letter. We identified U on that left-hand side, and we are in the row for that letter. Now, our second letter is A. So we come up here to where it says second letter. Here is A. So now what that means is you know which box your amino acid will be in. So we're in the U row. Now here, where I'm drawing this, it was supposed to look more red than that, but here is the box that we are in. Okay, so now we go to our third letter, which is G. So here's the tricky part. where it says third letter, you need to go to the row that you are in based on that first letter. So we are going to look at these right here. Now our third letter is G. It's right here. So we trace back to that box that we were in. Right here, you will find UAG. Now I screwed up because UAG is a stop codon and I did not mean to do that. But anyway, we'll just go with it. It says stop. So UAG is a stop codon. That's why I should plan out my sequence ahead of time. Okay, so we'll just write stop there for now. So now let's look at the second codon. I'm going to erase all this. Okay, our second codon is GUA. So we go to where it says first letter. Here's G. So that puts us in this bottom row all the way across here, right there. The second letter is U. So we go right here. We follow U all the way down to the row that we're in. And that should show you that we are in this box right here. Okay. Now our last letter for this codon is A. So you go over to where it says third letter. Here's A. Trace it into the box that we're at. And here you will find GUA, which codes for the amino acid valine. You do not have to know that VAL stands for that, right? You just need to know it's an amino acid, and we will write VAL right here. Now, when I ask you questions about the genetic code on the quiz or whatever, I will give you the genetic code table. You do not have to memorize it. You just have to know how to use it. So you should know how to go from DNA to RNA, messenger RNA, how to divide up your codons, and how to use the genetic code table to get the amino acid sequence. So let's do this last one here. Erase all my lines. Oops. Okay, so our last codon is CCU. We'll go through this a little bit faster. Here's C, here's C, here's U, which leads us to, oh wait, CCU. Yes, right here. CCU, and here is our amino acid proline. So we're going to put that right down here. Okay, so now we have used DNA to find the amino acid sequence. Okay, DNA to RNA, then we use a genetic code table to come up with the amino acid sequence. Okay, now that is only a part of translation. Actually, like I always say, a little bit more complicated than that. But that is the first part to you understanding how translation works. Okay, so in translation, there are 3 types of RNA that are used. Messenger RNA, which I just showed you. Okay, now I wrote the definitions here. Messenger RNA contains codons that determine the specific amino acid sequence. Ribosomal RNA, we also abbreviate that rRNA, that reads the codons on messenger RNA. And it also has 3 different sites inside of it, A site, P site, E site, and we'll get into that in a minute. Then we have transfer RNA, which we call it tRNA. Now that will bring a specific amino acid to the ribosome, and it also contains an anticodon that base pairs with the codon on messenger RNA. So let's see here. All right, so what you're looking at here is all of the types of RNA together. Now, we have messenger RNA right here. Okay, that is the strand of messenger RNA. It has the codons that are read by the ribosome. Now, the ribosome is made of ribosomal RNA. Remember way back from our cell structure lecture, how we talked about the fact that ribosomes are the site of protein synthesis. Well, this is what I was talking about. The ribosome attaches to that messenger RNA. It moves along the messenger RNA, reading codon after codon, and it signals transfer RNA to bring down the appropriate amino acid. So here we have a transfer RNA right here, and the transfer RNA will have a specific amino acid attached to it. Now, this is my slide specifically for transfer RNA. So transfer RNA, it's actually a single strand of RNA because all RNA is single stranded. It folds around on itself it kind of looks like a clover leaf. That's what they call it, a clover leaf shape. It's not actually a clover leaf shape. It's just kind of this bundle of RNA all folded in on itself. Now, each transfer RNA carries a specific amino acid. Okay, and it will bring that amino acid to the growing polypeptide chain during translation. Now, here is a tricky part about transfer RNA. It has what is called an anticodon. So remember, the codon is on messenger RNA, but that transfer RNA needs to temporarily bind with messenger RNA during translation. So the anticodon are the base pairs to the codon. So let's look at the picture on the right. Here we have I'm going to look at this transfer RNA right here. This is the transfer RNA I'm looking at. So the codon on the messenger RNA is UUC. Okay, that's the codon that is being read. The transfer RNA for that codon has an anticodon that is AAG, because U on messenger RNA base pairs with A on transfer RNA. U pairs with A, C pairs with G. So the anticodon to UUC is AAG. Okay, now notice here that this transfer RNA is carrying the amino acid phenylalanine, PHE. That is the amino acid that is coded for by UUC on the messenger RNA. So let's go back here. I know this is confusing, most likely, and I do have a video that I think will help with it. So let's go back to our genetic code table. We had UUC on the messenger RNA. So we have U, move my toolbar there, a U here. So we're in this first box. Here's our C, follow it over, UUC, and it codes for phenylalanine. So the messenger RNA codes for the amino acid. The transfer RNA brings that amino acid to the growing polypeptide chain. Okay. So let's go into a little bit more on ribosomal RNA, and then hopefully can all bring this together for you. I'm going to go back over here, actually. Now, I mentioned that ribosomal RNA has 3 sites. It doesn't look like I put a really good picture of it here. So what I'm going to do is I'm actually going to play this video for you during my video lecture here, because I want to build on it. So first of all, there are 3 steps to translation. They are the same names as transcription, but different things occur. So it's initiation, elongation, termination. So let's watch this video, and then I will talk a little bit more about it. Oh, hold on here.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, so let's continue here. Whoops. Okay. So I recommend that you watch that video several times to give you a way to visualize the process. So let's look at the first step of translation, which is initiation. So just like the video showed you, there is a strand of messenger RNA. Now remember this, messenger RNA is coding for a gene, right? It is coding for the amino acid sequence of a gene. We also call that a polypeptide. Okay, so here we have messenger RNA. Now, the first thing to happen is the small ribosomal subunit attaches to the messenger RNA. Now, a ribosome has a small and large subunits, and they are made of ribosomal RNA. So, okay, we got our messenger RNA, the small subunit attaches. It starts to move along that messenger RNA until it hits what we call the start codon right here. The start codon is AUG. Now, the codon AUG codes for the amino acid methionine. So most proteins start with the amino acid methionine. So I'm going to write this down for you here. Oh, I have it written. So ribosomal subunit binds to messenger RNA, and the start codon is AUG. So once that small ribosomal subunit hits AUG, the transfer RNA or tRNA with methionine binds to the AUG codon. Now, that transfer RNA has an anticodon, UAC. That's base pairing with AUG. Okay, but remember, it is always the codon that specifies the amino acid. It's not UAC that specifies that amino acid, it is AUG. If you use UAC on the genetic code table, you will not get methionine. Okay, so transfer RNA comes down with methionine, base pairs with AUG. Now the large ribosomal subunit joins the small subunit to make the full ribosome. Okay, so I'm going to repeat that again. Messenger RNA, small ribosomal subunit attaches, moves along the messenger RNA until it hits AUG, the start codon. A transfer RNA with methionine base pair and binds with that start codon. Then the large ribosomal subunit attaches, making the full ribosome. Now that initial transfer RNA with methionine sits in the middle of the ribosome at the P site. It's important that you remember that. Now the video only showed an A site and a P site, and that's fine. Some sources show an E site, some don't. Now we are moving on to elongation. This is the easiest to understand image that I could find for elongation. And then I summarized it here on the left. So that initial transfer RNA with methionine is in the P site of the ribosome. Now the A site is open, and there it is over a codon. That codon specifies the next amino acid in the sequence. So a new transfer RNA with that amino acid comes down and enters the A site. The amino acid on the transfer RNA in the P site transfers over to the tRNA in the A site. We call that translocation. Okay, so amino acid goes from the transfer RNA in the P site over to the transfer RNA in the A site. The ribosome moves down one codon. So the transfer RNA that's in the P site, and it's now empty, it has no amino acid, it leaves. The transfer RNA that was in the A site is now in the P site, leaving the A site open. A transfer RNA with the appropriate amino acid comes into the A site. The amino acids that are on the transfer RNA in the P site, they move over. The ribosome moves down one. The transfer RNA in the P site, which is now empty, leaves. The transfer RNA that was in the A site is now in the P site, leaving the A site open. And like I said, watch the video numerous times until this process of elongation makes a little bit more sense. So if we look at the picture here, we have AUG right here, that's our start codon. We have a transfer RNA with methionine that is going to attach to the start codon. That is in the P site. So here we have that left UUU as the next codon and a transfer RNA with phenylalanine, which is what UUU codes for, enters the A site. Now, here's the part that's kind of tricky that everyone gets a little confused on, but the amino acid that was on the transfer RNA in the P site, it moves over, right? So notice now in this last picture, the transfer RNA has phenylalanine and methionine because that methionine moved from the one tRNA to the other one. Okay. Now the empty transfer RNA will leave the ribosome. That's this guy right here. It's going to leave the ribosome. Okay, so now the transfer RNA that was in the A site is now in the P site, and the A site's open. That is where another tRNA will come down, and the amino acid sequence or the polypeptide will grow larger, larger, larger. That is the process of elongation. Now, the last step in translation is termination. This is where the stop codons are important. So the ribosome is moving along that messenger RNA. It's reading codon after codon after codon. Well, eventually it will hit a stop codon. Remember, there are 3 different stop codons. When this happens, the polypeptide, the amino acid sequence is released. The ribosome comes apart, small and large subunits, and the messenger RNA is no longer attached to those. That is the end of the polypeptide, the end of the protein. It goes off, and then it will be folded so that it is a functional protein. So you have initiation, elongation, termination. I wrote this slide with key points to remind you of the information that you need to retain. So for transcription, the location in the cell is the nucleus. The function is the production of messenger RNA from DNA. I'm going to write these down for you here. So location is the nucleus. The function here we go, is the production of messenger RNA from a DNA template, right? So you need to remember DNA to RNA base pairing. So refer back to the chart that I made you. So I'm going to write the DNA bases first. So A pairs with U, T pairs with A. So that means T in DNA pairs with A in RNA, and G pairs with C. C pairs with G. Okay. The function of RNA polymerase is to add RNA nucleotides to the growing strand of RNA. There we go. Show it as RNA nucleotides to the growing strand of messenger RNA. And then finally, RNA processing. This occurs in the nucleus right after transcription. So I'm going to write occurs in nucleus after transcription. And there are two main things you need to remember. Cap and tail are added to RNA and the introns are removed. Remember, the exons are the coding region, the region that is needed to make the protein. So I have a summary for translation. So key points. Translation occurs in the cytoplasm. The function is the production of a polypeptide from messenger RNA. I'll put synthesis. Remember, a polypeptide is the sequence of amino acids from information in messenger RNA. When I write that it's a synthesis of a polypeptide from information in messenger RNA, I mean codons. So you need to know how to use the genetic code table. So you are going to use codons to determine a specific amino acid. So I'm not going to summarize functions of messenger RNA, ribosomal RNA, tRNA. You need to go back to the slides for that because I don't have enough room to write on that. Also, you need to know what happens during initiation, elongation, termination. So there are also some other great videos for translation. My favorite, the Amoeba Sisters, is out there. There are other videos like Crash Course. So I recommend that you utilize these resources if you find that you are really confused on transcription and translation. These are a bit confusing. They are complicated processes. You can also reach out to me and we can go over this in a Zoom meeting.